In bacteria, there are three major types of RNAs, that are mRNA, tRNA, and rRNA, that is ribosomal rRNA, which is for synthesis of protein. Now, but all these three RNAs are needed to synthesize a protein in a cell. So all these three RNAs, three major types of rRNAs. In bacteria, there is a single DNA dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria, that is mRNA, that is messenger RNA, then there is tRNA, that is transfer rRNA, then the ribosomal rRNA, rRNA, right? So a single DNA dependent RNA polymerase. What is transcription? So transcription is a process. What is transcription? Transcription, the process of copying genetic information. The process of copying genetic information from one strand of DNA into RNA, the mRNA into mRNA, is termed as transcription. So this is transcription. Now, so we have seen the types of RNAs in bacteria. These RNAs are transcribed by a single DNA dependent RNA polymerase. Okay, single DNA dependent RNA polymerase. Now, but in eukaryotes, so this was in the prokaryotes, but in eukaryotes there is two additional complexities. The first one is there are at least three RNA polymerases. This is the first complexities. In bacteria, it was single DNA dependent RNA polymerase that transcribes all types of RNAs needed for the protein synthesis. But in eukaryote, at least three RNA polymerases in the nucleus, in addition to the RNA polymerase found in the organelles. So particularly these three RNA polymerase found in the nucleus. Now talk about each RNA polymerase. So there are at least three types of RNA polymerases. So the RNA polymerase 1, what is the function of RNA polymerase 1? So RNA polymerase 1 transcribes its copy. Transcription is the copying of genetic information from one strand of DNA into RNA. So RNA polymerase 1 transcribes rRNA, that is ribosomal RNAs. Okay, these rRNAs include the 20S, 18S, and the 5.8S. So these are the rRNAs that transcribed by RNA polymerase 1, whereas the RNA polymerase 3 is responsible for transcription of tRNA, 5S rRNA, and SNRNA. RNAs. So this is the function of RNA polymerase 3, the transcription of tRNA, 5S RNA and snRNA. snRNAs, these are small nuclear RNAs. Now the RNA polymerase 2. So RNA polymerase 2 transcribes precursor of mRNA, that is the heterogeneous nuclear RNA, that is hnRNA. So RNA polymerase 2 transcribes precursor of mRNA, the heterogeneous nuclear RNA, that is hnRNA. So in eukaryotes, two additional complexities. The first is at least 3 RNA polymerase 1, RNA polymerase 3 and RNA polymerase 2. Now the second complexity is the process of splicing that converts the precursor of mRNA, that is heterogeneous nuclear RNA, into mRNA by capping and tailing, capping at 5' end of hnRNA and tailing at 3' end of hnRNA. So splicing involves capping and tailing. So this was the second complexity. So in bacteria, only a single DNA dependent RNA polymerase transcribes all types of RNAs required for the protein synthesis. And in eukaryotes, at least 3 RNA polymerases are needed. So RNA polymerase 1, RNA polymerase 3 and RNA polymerase 2. And the second complexity is the process of splicing that converts the precursor of mRNA into, that is the hnRNA, into the mRNA.